Welcome to another edition, in fact, the final edition for the year of the NDF Speaker Series. And today we are honored to be joined by Dr. Francis Rossignol. Just to give you a background, Dr. Rossignol is a medical geneticist who trained in Canada and came to the US in 2018 upon receiving a prestigious fellowship at the NIH from the National Human Genome Research Institute. So today he's invited to give a talk on the ManNAc trials. He's a principal investigator at NIH for GNE Myopathy, where he heads the natural history studies, as well as the ongoing ManNAc trials, and is also one of the co-principal investigators at the Multicenter MAGiNE trial, which I'm sure you're all familiar with, that is sponsored by Leadient. So without further ado, I want to pass the stage on to Francis Rossignol to give us an update about what's happening in the ManNAc world.
Well, thank you very much for the kind introduction. So I'll start the slides and share them. But yeah, as I said, my name is Francis Rossignol. I'm a biochemical geneticist at NIH. And I always say that I'm the lucky person that gets to talk, but most of the work that I will be presenting today has been done by NIH throughout the last 20 years. So I have a lot of very dedicated colleagues that led those efforts. I'm the just a lucky person that gets to talk today and head some of the more recent work that we are doing with ManNAc and GNE Myopathy. But certainly I want to make sure that everyone realizes how much of a team effort this is. Was asked to incorporate a summary slide. So just to give an overview of what we'll be talking about, ManNAc is a non-charged precursor of sialic acid. So the goal is to supplement sialic acid in the pathway. We'll talk a bit more about it, even though pretty sure that many people are already familiar with the pathway. And in a nutshell, we've seen in the past that the administration of ManNAc in individuals with GNE myopathy increases sialic acid production, both in plasma and in muscle. And then we have some data that suggests clinical efficacy, but further studies were needed, and that's where we are at the moment. And we do have an ongoing study recruiting. So I'll skip about this one and go straight to the pathway. So I'm sure that you're familiar with the pathway. You've seen that a lot, but there's a couple of things that I want to highlight because it's fairly important to understand some of the characteristic of ManNAc itself. So that's the sialic acid biosynthetic pathways, which is deficient in GNE myopathy. GNE has two roles in the pathway, so both an epimerase role where UDP-glycanac becomes ManNAc, and then the phosphorylation role, which is the ManNAc kinase function that brings ManNAc into ManNAc 6-phosphate. And then ultimately, all these precursors go to sialic acid that are used to sialate proteins that are important in many different processes. We'll skip about all the names and the prevalence. You probably heard that many times, but it's a you know that it's a fairly rare disorder with about 1 to 9 in a million. The exact pathophysiology is unknown, so there's a good deal of mechanisms that are being explored, and I know there are some people that did talk about in the series about some of interesting new pathomechanisms that are being explored, but one of them that has been thought since the beginning is that the hyposylation of muscle proteins is probably involved. And then many different precursors of sialic acid have been explored, but none have reached approval so far. There's a natural history of the disease that we are studying. It is important to understand it to be able to better see the clinical outcomes that are related to whatever intervention that we're doing. So we know that in typical fashion. It presents more in the distal limbs, distal lower limbs first, then progress proximally, sparing the quadriceps, with an age of onset between 20 and 40, but there are substantial interfamilial variability, which is a challenge for any clinical trial. And then we do know that the anterior tibialis, so the ankle dorsiflexors, are usually the first muscles to be involved with the foot drop, the gait disturbance, but then the rest of the pattern is not necessarily always the same for everyone. So this is also another challenge where we know that upper limbs, what we've seen in our natural history is that they're typically involved 5 to 10 years after onset, but they don't necessarily follow a pattern from distal to proximal or proximal to distal. So it's a bit less predictable, which is another challenge that we had to deal with. And with time, there's more progressive reliance on assistive devices, with many people being in a wheelchair between 10 and 20 years after onset. So that's what we gather from our natural history, some of the data that we had in there. They're important to know, but there's obviously some variability in there that we need to address, and I'll talk a bit more about this. So for those of you who are curious of the biochemical formula, here's ManNAc itself. So it's been identified as a potential therapy. You'll realize that here we have the two functions of GNE, so the epimerase domain and the kinase, and it falls in between the two, and that is one of the challenges we will address. But one of the important thing about ManNAc is that it's uncharged. So many of the molecules, actually all the other intermediates in there, are positively charged. They do need some mechanism to enter the cells. The advantage of having an uncharged molecule is that it can go in more freely. So that's why it was thought to be a very good option, because it didn't rely on a transporter, for example. And we do have some preclinical data. So I'll have a couple of slides on this. You may have heard of this mice model. They are not necessarily fairly new, but there's both the NIH group and Marianne Husing has been involved in that model, and the Nishino group, the second model where Mimalik Den, who's also now at NIH, has been involved. Both of them have shown some significant effect of ManNAc supplementation. So both increased survival in the M712T mice, where we can see that with lower dose of ManNAc, we do see more survival of homozygous mice, and we do see some increase in silation in muscle, for example, where the homozygous mice, when they're untreated, barely have any silation, as shown with SNA lectin staining, whereas once we administer some ManNAc, this muscle silation is coming back towards more normal levels, which is encouraging. And the Japanese group mouse model did show similar effects, so where we see that higher dose individuals are actually undistinguishable of controls and healthy litter mates in terms of survival, and they do have some improvement of their outcome measure. So we can measure, for example, the distance that they run, the degree of muscle atrophy with the muscle weight, and we can see that it is significantly different from the controls that aren't treated, and it goes back to towards the normal levels that we would be expecting. But the key question that any clinical group has to ask themselves when it comes to therapy is, how do we go from something that's promising in an animal model to an approved therapy? We all know that the mice models have limitations. They're not acting necessarily in the same way than GNE myopathy is acting in humans. So we know, for example, that some of the mouse model have kidney disease, which is not reported to be associated. So there's obviously some limitation. There's some caveats to it. And so we need at some point to study it in humans, but how do we achieve that in a disorder where there's some substantial variability? So, and how do we manage to convince everyone that the therapy that's being proposed is valid and has a good effect? That's another challenge. And I like to illustrate this from a chart. That chart comes from actually the HIV website from NIH. So it is how more typical trials are being conducted. And we will soon see, and probably you are already noting in the slide some issues that this is unachievable for a rare disorder. So the goal of any clinical study is to go from preclinical data like the mouse model and then go towards different phases where safety is going to be examined throughout the process. So early on, it's only safety. As we go, we're looking more and more into effectiveness. So we may have some effectiveness data in phase 1, which is limited, and then biochemical effectiveness or some mild like attempts at clinical outcomes in the phase 3, and then phase 3 really focusing on effectiveness and clinical outcomes to then go to an FDA review for approval of the said drug, and then phase 4, which are the post-marketing studies where it's more long-term safety and effectiveness once the drug has been approved. But obviously, there are some challenges in there. So one of the first one is, what do we know about the condition? What do we have a therapeutic target? Here we have several therapeutic targets. We know the gene, we know the pathway that's being involved. So there are several therapeutic targets. What do we know on the condition? That is a bit more of an open question sometimes. So it depends on how much data we have, and that's where the natural history studies come in handy. And how do we measure effectiveness? So it needs to have an understanding of what is disease progression, what are we expecting to happen, and how can we measure the change in time? Is it uniform progression, or is it more variable, which is another challenge? If it's more variable, we need some ways to normalize everyone so that we're able to compare people the same way, basically. And then do we have biomarkers? Do we have clinical markers that are sensitive enough to detect the change in the duration of what we're looking at? Then there's the big question, which is probably the most obvious one from this schematic. How many people will this require? Like you can see here that for the HIV trials, they mentioned that phase 3 have 1,000 to 3,000 participants. This is unachievable in rare disorders. So how will you find enough people? How can we detect a significant change in a minimum number of participants that is still significant and will still convince everyone that we are effective, but still manage to gather with the fact that we have a limited number of affected individuals and there's only so much that we can do. And then where is the funding coming? Where's the expertise, which is also another challenge in any rare disorder, where expertise is usually concentrated in a few limited areas, not necessarily as broad. So you cannot necessarily have the multi-center trials with as many centers that other trials that recruit 3,000 people will have. So there's many challenges and many ways to address those challenges, some of which we've taken. So what do we know on the condition, and the basic science help us, but how to measure effectiveness? We use the natural history studies to know more about disease progression, know more about how we can try to have comparative measurements that we can bring to a clinical trial. Then on how many people, both the development of outcome measures play a role into that, but alternative study designs and how many people will be on the drug, how many people on the placebo, how many centers you're going to involve, how many, what is your primary outcome measure, all these play in your power to detect a significant change. And then obviously for the expertise, it involving most of the stakeholders. So we need to make sure that both scientists are on board, patients are on board, families are on board, everyone needs to be involved to be going through the entire process. So from that, I'll transition more to what we've done with natural history, and then for the phase 1 and phase 2 of ManNAc and NIH. So most of that comes from our NIH data. So for those of you who are not familiar with the natural history study, so we do have a study where we bring people at the NIH that's a picture of the NIH Clinical Center, which is where my office is. People come for 4-day admissions, most typically. Initially, it was every 6 months. Now we've spread it out a little bit more. It is more, I would say, at a ad hoc basis, given that we have a lot of people coming on the clinical trial. In total, we've seen 69 different individuals. 56 people came to NIH for the full evaluation, many of them multiple times, which was a tremendous help, and we could not have achieved it without all these people coming. And there's 13 people that participated in mostly phone interviews and questionnaire development, where questionnaires were sent at home for development of new outcome measures. And that's to give you a bit of an overview of what we would do over 4 days. So the Monday is mostly like admission and getting a good history, good physical exam. And then we do quantitative muscle assessment, which I have a separate slide just after, because that's a central measure for our all our trials. So I want to spend a bit more time on this. And then we have a day of lab, other physical measurements like the 6-minute walk test, manual muscle testing, adult myopathy assessment. We do some cardiac evaluation. We know that most typically there is no cardiac involvement in GNE myopathy, but when we were in a natural history, we were looking for everything that could happen, even though we think it doesn't happen, or it has not been reported or not reported frequently. So we do investigate a bit more aggressively, I would say, to try to find milder outcomes that may relate to the condition and were not known previously. Given that the heart was a muscle, we did look at everyone on that front. And we do the same with pulmonary function tests, for example, on Wednesday, which is where we most typically have them. So we're looking if there's any evidence of it, which is not usually a concern. We do have some occupational therapy evaluation where it's more fine motor skill evaluation. And then on the Thursday, wrap up of all the summary of all that happened during the week. One of the main outcomes that I wanted to come back to is a quantitative muscle assessment, or QMA. So this is a device. I usually say that when you look at it like that, it does look like a torture device. Trust me, it is not that bad. I've went in that machine several times. I'm still okay. And I wasn't okay before, and I'm still good. So the principle here is to try to measure muscle strength as in raw numbers. So we need to have some objective measures to bring to clinical trials, and that's one of the methods we've used. So basically, we have a load cell, which is attached to the frame, and you can see on the right that there's a strap that is brought to the limb. And once the person here will be asked to kick out their limb, then the load cell that's at the end of the strap will register the strength, and then it goes on the computer. It's only for 5 seconds. We take the max, and then we compare it to a database of normal data, which we can give a percentage of predicted strength depending on height, weight, age, gender, so on and so forth. So that really help us define which are the main muscles that are involved, in which fashion, at which rate of progression. In the natural history, for example, we were studying 13 different muscle groups, including many in the upper limbs, the lower extremities, including all the hip flexors and ankle dorsiflexion and knee flexion, and so on and so forth. So that longitudinal data was really instrumental to what became our main outcome measure. And that's some of the data that has been published now 3 years ago, but it relates to what I was saying earlier, that we do not necessarily know how there's a variability in between families. There is a variability even inside a family with disease onset and rate of progression. So you can see here when we plot muscle strength as percentage predicted, so 0 being 0%, 1 being 100% of predicted, but you can see that over chronological age, it doesn't seem to make any sense. Like, it's just a scatter of points that are not, they are not regressing to a linear model. Even like late, we have some people that have fairly preserved grip, for example, whereas some early on do have fair amount of grip strength loss. So it is very hard to use chronological age as a predictor of where someone should be in terms of their disease progression. But in this figure, there's also some people have been labeled with A, B, C, D, and those are single individuals. So we can see that in a single individual with a chronological age, for example, in shoulder abduction, it tends to trend down with time. So if we look at individual level data, it does trend well, but when we try to gather everyone together, that's where it does not hold up, and it is a major problem to bring to a clinical trial. So with the help of our statistical consultants, with some modeling, that we came up with the GNE Myopathy disease progression model, which has been designed to become a new outcome measure. And the way it works is that we try to normalize everyone on one muscle group. So basically, once knee flexion is at 50% of predicted, we call this age 0. And that becomes the new baseline for everyone to try to compare themselves to the others. So we get rid of the chronological age that did not correlate, and then we bring everyone on a new age, which we'll call the disease age. But there's several parameters that were inputted in the model, including chronological age, muscle variability, timing. And then once we get that, I added those lines to kind of give an overview of like when we are at 50% of knee flexion, which is where we define age 0, which is the center of those two lines. Then we can look in terms of disease age on the horizontal axis and see a bit what we would predict in terms of progression. So for example, when someone has 50% predicted of their knee flexion, we would expect that about 7.5 years earlier, that's when they were at 50% of their ankle dorsiflexion strength. And that is when 10 years later, that's about when we would expect hand grip to be at 50%. So now we are trending towards being more able to predict a bit more what is actually happening. You can see that there's shaded area around every of those lines. That's kind of the error rate that we do have. So because not everyone is going to Google at the same rate. So there are some places where you can see that it's much larger and we have a bit more of imprecision on our prediction, but towards we have a general pattern where it plateaus at the beginning, it does progress, and then it plateaus towards the end. And that's kind of our general model of progression. So that has become very handy. And then what we did is to compare it to the other elements that we were gathering through the natural history data. So we have, yes, the raw strength data, and those are the three first ones, that the three first lines here in terms of total strength, either upper extremity, lower extremity, or total. And then we have all the other functional measures like the 6-minute walk test, the adult myopathy assessment test, the HAP, which is the human activity profile, which is more a questionnaire of 90 plus activities and whether or not people can still do it or not, or they have more difficulty doing, or the inclusion body myositis scale, which is another of the questionnaires that we're giving. And we can see that with chronological age, some of them do correlate. So the human activity profile does correlate with chronological age, but most of the outcome measures that we were looking at in terms of chronological age were not correlating well. Same goes with disease duration, which is actually a better predictor than chronological age. So we were always asked to everyone, when did they start noticing their first manifestations of it? So we have an idea of how long it's been since they've experienced muscle weakness. And this is a better predictor actually than chronological age, but not perfect. So even like for strength, it does not correlate well, but we do see that it correlates better with the 6-minute walk test, for example, than chronological age. But when we come with our disease progression model, we can see that all the measures here are all very significantly and highly correlating with the disease age. So the more advanced in terms of disease age someone is, the more we can, like, their 6-minute walk test will go down, and their AMAT will go down, and their total strength will go down. So we have here a much better predictor of not only raw strength values, but also other clinical outcome measures, including patient-reported outcomes, which is an important part of what we want to look at, and regulators want us to look at. So FDA always says, well, it's good to have the number to show on something, but you also want to see that you're actually making a difference. So to be able to show that, yes, our outcome measure correlates with some scales that are designed to address more of the day-to-day and reality of the disease, and not just the raw values of how many kilograms can someone grip, is very, very helpful, actually. So that's where we're very happy about how the model is performing overall. So this is an example, and some where we see someone that's more earlier in the progression, where the hand grip, so the green areas are more preserved, the red areas are more affected, and the orange ones are a bit midway. So we're here at someone that's disease age 0, where the knee flexion is at 0. So that some of the posterior muscles here are in orange, but the quads and the hands are relatively preserved at this point. And then we can see that ankle dorsiflexor are much affected. And we can see here that some of the items of the IBM-FRS, which are like, it's probably very small on your screens, but here it says like capacity to dress oneself and handling utensils and walking and risk of falls. You can see that early, most of these are in a good zone where it's not really affected, except for the risk of falls, which is trending toward yes, probably because of the ankle dorsiflexion weakness, but most of the data is more towards the upper limit of the scales, the better outcomes. And then once we progress and we see that actually there's more muscle involvement, and we can see here that most of the muscle groups are now in red, and even the quads here are involved in some fashion. So here it'd be like about 70%, probably like 65% predicted for the quads. And we can see that many of overall all the items on the IBM-FRS of how people are faring as terms of climbing stair, walking, dressing themselves, tends towards lower scores. So there is some correlation between the muscle outcomes and the raw values that we're seeing on QMA and normalized across disease age and other clinical outcomes. The other thing that was developed with it is the treatment parameters. So and it becomes very important to understand that concept for the future clinical trials and what we're doing at the moment. So we did incorporate what we call the gamma parameter, which once a treatment is initiated, we can compare what we would expect as the natural history, being the normal progression that we would expect without an intervention, which is the solid line. And then we have different gamma, where basically a gamma of 0 means that we've completely halted the progression and the muscle is completely stable in terms of strength. And then once we're at a gamma of 0.5, it's basically a 50% reduction in the rate of progression. And if we were at a gamma of 1, basically we did not have any effects on the disease. But we can see that there are some challenges in there, where in the figure A at the beginning, so we're probably at here treatment that's initiated at disease age -5. And then if we want to see a significant effect, like in the first few years, the rate of progression is fairly slow. So it's hard to make the difference between something that has not progressed and something that was towards the natural history trend. But with time, it does affect tremendously. So we see that we could make a difference after 15 years very clearly, but in a short time frame, much less so. Whereas if we are more towards the end of disease progression, where we're at disease age that's more advanced, then it is difficult to make the difference between the natural history, which would be the solid line, and the treatment ones, because there's not as much sensitivity. So our measure, even though it correlates well, has some limitations in terms of the sensitivity to detect mild changes, for example. So, and in which time frame mostly. So it becomes very critical for us to recruit and enroll people that are more in the active progression of the disease, where it's the slope is the most acute, so that in a short amount of time, we're able to address the question of whether or not a treatment has some efficacy on muscle and not take 15 years to come to approval, which would be detrimental to everyone. And that's only part of the work that we've done. So I've centered it on QMA a lot because that's what we're studying most with the current trial that we're ongoing, but there are several other outcome measures that became very important with the earlier phases. So for example, the development of SNE-lectin staining in muscle, where here we have some, we can see the blue, which is some the SNE-lectin that's capturing, and then we have the red, which is a control, which is caveolin 3 in this case. And this is a sample of after treatment where we see a bit more muscle with SNE-lectin staining. So that was used during phase 2. I'll show some of that data a bit later on. But we also studied, for example, how muscle mass and fatty infiltration in muscle using spectrometry was correlating with other measures. It is a much harder thing to obtain everywhere, so it's not a measure that we currently are measuring in the clinical trials, but there is some correlation between muscle MRI and clinical outcomes, including IBM-FRS, ABC scores, which are two different scales that we're using, the 6-minute walk test, the upper and lower body strength. So we do see correlations on these fronts as well with other things that we've performed through the natural history. Another big part of the natural history, and that actually stems even beyond natural history, it was for us to review all the available data in terms of variants. And we have seen several variants that are unique to families. We know that many variants in a GNE myopathy are missense variants. So there are changes in the gene that will cause one building block of the protein, one amino acid, to become another, but not necessarily cause termination of the protein. So the protein totally not being produced. So we see a lot of those milder, quote unquote, genetic changes, which are harder to give an interpretation on, because once they're seen in many individuals with the disorder, it's much easier to say that, oh yes, it really is associated with the disease. Once it's been seen only in 1 or 2 individuals, it takes a lot more evidence than if we had a big top codon in the middle of the gene. We would say, oh, that's more likely to cause a problem. So we do have challenges in interpretation because of that particular fact of how the gene is affected. So we are carefully reviewing all the work, and this is a figure that Marianne Husing put together that I really refer to it almost every week, where we look at the variants, we look at how much data we have on each variant. Do we have other studies? Do we have in vitro evidence? Do we have how many people have been reported with these variants? And we accumulate more and more data also through the natural history. We do some extra testing in our research lab to bring more evidence. And it became very critical to do so, because once you come to the clinical trials, you want to make sure that everyone that's coming has variants that are convincing enough that you don't think that there's any possibility that it could be another disorder, which is sometimes frustrating for some, because we are pretty sure when we see the variants that it is very much likely it is GNE myopathy, or it is GNE myopathy clinically. And we know it, but we are not able necessarily to gather enough evidence to for the genetic variant. So we are working very hard on getting those variants solved as much as possible and gather as much information as we can, so that we can present a cohort in the clinical trials that is as solid as possible. And then here comes some of the data that we accumulated through phase 1 and phase 2. So phase 1, as I was saying earlier, phase 1s are all about safety. And so we're mostly interested in measuring, does ManNAc increase sialic acid, which is our endpoint. We wanted to see whether or not we were causing the change that we want to see at the end in terms of biochemistry, and how well tolerated it is. So it was a single dose placebo-controlled trial where people were either given placebo, which at the time was mannitol, or 3 grams, 6 grams, or 10 grams of ManNAc in a single time frame. And then we would measure both ManNAc in blood and sialic acid to show that there was some efficacy in restoring sialic acid production. And overall, from that data, we could say that in these individuals, it was well tolerated. The higher doses tended to have a bit more adverse events, so a bit more of diarrhea and nausea in the 10-gram group, which we attributed to unabsorbed ManNAc. So ManNAc is a sugar. Sugars in the digestive tract that are not absorbed tend to bring some liquid and by osmosis, so there's some more water in there and will soften the stools, and then the bacteria can feed on it and release gases, for example. So it's probably what is happening there, that it's not fully absorbed and causing some issues. We also observed some elevations of liver function tests, but in a mild manner, nothing that was really a concern clinically. It was more a biochemical finding by itself. It also allowed us to know how ManNAc is acting in blood. So it has a very short half-life. We can see that it peaks after about 2.5 hours. And after 2.5 hours after, we're basically at half we were. So it goes away in the blood fairly quickly, which suggests that repeated administration might be better. But then the sialic acid innovation following ManNAc administration is actually much longer. So we can see that it peaks here in blue more towards 8 to 11 hours after administration, so much later. And then when we stop examining all the samples, which was about 40 hours and 40 or 48 hours after administration, we can see no more ManNAc. We're back at baseline, but we still see at that point sialic acid innovation. So even a single dose of ManNAc results in a longer term sialic acid elevation in blood, which is very encouraging in the long term. So with all that data and what we learn about half-life and the pharmacokinetics of the drug, it suggested that the dosing of 6 grams twice daily may be the most optimal. So that came mostly because the 10-gram dose did not increase sialic acid more than the 6-gram dose in proportion. So we can see like the squares here are the 6-gram dose, the 10-gram dose is in with the triangles. With ManNAc, it does increase tremendously more. But when we come with the triangles and squares on the sialic acid front, we basically see the same thing. So we're probably limited in terms of the absorption at that point, or the capacity of the cell of turning ManNAc into sialic acid more. So we do see some significant limitations on very high doses, even though they're tolerated. So that's with that model that we came with the phase 2, where the goal was more to show a closer to clinical endpoint. So the main endpoint in the phase 2 study was to show biochemical efficacy in the muscle. So we've seen 12 people that were enrolled in 2 groups. So there were no placebo groups on this one, but either a group that started on 3 grams of ManNAc, another group that started on 6 grams. And after 7 days, everyone was put on 6 grams of ManNAc. It was to study some of the tolerability. And then after 90 days at day 0, they would get muscle biopsy with MRI guidance, and then 90 days later, a second muscle biopsy just next to the first one with also MRI guidance to make sure that we were as close as possible and comparing an area of the muscle that was similarly affected. So we were selecting the area on muscle that showed hyperintensity and suggested that it was actively changing at the moment. So we were more likely to find something that would cause a difference. So at 90 days, then we would compare the two. And then there were study extension where some people stayed on the drug as much as 30 months after, where we had more of the safety data on the long term and some of the clinical outcome measures long term. So the primary outcome measure, as I was saying, was the muscle biopsies and the lectin staining, and so the contents of sialic acid in the muscle. And then we had secondary with the QMA and some of the other assessment, both physical, like the 6-minute walk test and the AMAT, or the patient-reported outcomes like the IBM-FRS and other scales. So it showed overall good tolerability, a bit like we've seen in the phase 1 with single doses. There were some people that did have they report gastrointestinal distress from it, which is the main issue that has been since the beginning. But towards the end of the study, the regimen was changed from a 6 grams twice a day to a 4 grams 3 times a day, and that has shown better tolerance overall. So we are using from now on 4 grams 3 times daily instead of 6, which is the same total dose in the day, but more spread out. And here's some of the data that we've seen. So, for example, that's muscle here at the left at baseline, where we can see a lot of red, which is a caveolin 3 that we use as a comparative marker to see that our staining worked. And SNE-lectin is the sialic acid marker that we want to look at. So we see that there's some silation in the muscle, but obviously we see a bit more red than we see some of the blue, which means that most of the muscle here is desilated and doesn't have a lot of sialic acid. And after 90 days, we can see a lot more of the blue in the muscle. And it is not only your eyes that are tricking you, because that could always be the question. Is it just an effect of us being unblinded and knowing that we're expecting that? But we also took pictures and quantified the intensity of staining, and we can show that compare from baseline to the 90, overall, there's an increase in the SNE-lectin intensity, even when normalized to caveolin 3. So there's good evidence that administration of ManNAc not only increase sialic acid in blood, but does restore some of the silation in the muscle, which is the most important thing that we wanted to look at in the study. Another point that we wanted to check, and that comes back to the biochemical pathway and why knowing the pathway was important. So we did look at how ManNAc would increase sialic acid levels in all individuals, and depending on their genetic variant status. So as I said before, GNE has 2 functions. There's an epimerase function, which is responsible for transforming glycanac, UDP-glycanac into ManNAc. Then the kinase function that's responsible for adding a phosphate to ManNAc and create ManNAc 6-phosphate. So someone may tell me, well, you're giving ManNAc, but then the function after is affected. How are you expecting the ManNAc to go to ManNAc 6-phosphate and progress in the pathway? So we've shown with the previous phase and that ManNAc still increases sialic acid, despite the fact that probably kinase function is deficient, but we're still seeing some increase in the end product. And that probably relates to the fact that there's other kinases that are present, or some residual function of GNE as well. So both of these may contribute to the fact that we're still seeing increase in sialic acid. But we wanted to address that question more specifically, and with more people, we were able to address that. And you can see here on the right, it's not a figure that if you've been interested in the literature, that this comes from the supplementaries of the phase 2 paper we've published. And most people have epimerase and kinase domain variants, so a bit of mix of the both functions. But we did have a couple of people that were having kinase domain, 2 kinase domain variants. And we can see that sialic acid through time did increase with repeated doses. So the red one was at the beginning of the study, and then towards the end of the study, we are more in the blue and the green. And we can see that even with 2 kinase domain variants, we're still seeing a sialic acid elevation, which could be a worry. Right? If we say, well, in theory, the function here between ManNAc and ManNAc 6-phosphate is completely gone, we could not see sialic acid, but the evidence we have is that it still increases sialic acid, and we're optimistic that it's not a medication that will be, for example, variant specific, or that will need to target only some people that have some variants but not others. So far, we're seeing an effect on everyone. The study was not necessarily powered to show clinical significance on clinical endpoints, but we still saw some very interesting trends. So that's some of the data compared to natural history. So there's always a reserve when we come to regulators with natural history data, where they'll say, well, they've not been studied at the same time. So bear in mind that this is compared to a historical cohort, prospectively collected through our natural history, but still historical in terms of, and not studied at the same time. But we can see for upper and lower extremity strength in both A and B that basically are the individuals, the 12 that were on ManNAc, overall stabilized their muscle strength compared to baseline, whereas in our natural history, we would have predicted change from baseline that was much more significant. And we can see the p-values are highly significant. We would taking a cutoff of 0.05. We are way below that for both of them. You can see that there's a couple of missing points in the upper extremity strength in B. That's because the hand grip dynamometer that we were using at the time just stopped working, and it took some time to realize. And it was not working properly, and the data was not reliable, so we have lost some data there. But that has taught us to be very careful of our equipment and calibration some more during the current trial. So we're much more aware of this now. But we can see as well that some of the other measures that we were looking at, like the 6-minute walk test, we didn't see any difference. The HAP, which is and the IBM-FRS, we don't see any significant difference. So there are some other outcome measures that we did not see difference. Limitation being that with 12 individuals compared to a natural history, we were not powered to see those differences. It takes more people to be able to address those differences more clearly. So that's certainly some encouraging data on the muscle strength, but we still had work to do with that. And I wanted to come back to the treatment parameter, because we did try to see if we had a treatment effect using our disease progression model. And we can see that here, a gamma of 1 is what we want to avoid, actually. So when we're not overlapping with 1, that's what we want to see. At 24 and 30 months, because of the loss of the upper extremity, we are not in the significance, but at 12 months, we see that probably the gamma is towards like 0.6. The range does overlap 1, so it's not quite significant. Same at 18 months, where we see that the probability of gamma being less than 1, which is what we are aiming for, was 96% or almost 97%. So very encouraging that it does slow progression, but not quite powerful enough yet to be able to show a significant difference. So that's where the current trial is coming in. And you're probably aware this is an ongoing trial. We're just currently in the process of getting our first 6-month visits data. It is a blinded study, so I do not have necessarily data that I can share that will tell you, oh, here's how it looks in between the groups, because I'm totally blinded and I need to remain blinded for the duration of the study to not skew any measurements. But I can provide a bit of the general information on what we're doing. So it is a multi-center trial. As Dr. Lex said, initially we have a pharmaceutical partner with us, which is Canadian Bioscience. We have NeuroNext, which is a network that helps us coordinate with other centers in the US. So NIH has done some of the work previously. NeuroNext is helping us having much more centers and coordinate between all those centers. So there's multiple centers that are currently enrolling. You have the list here. And so between Brigham and Women, Columbia, Ohio State, UCLA, University of Iowa, Rochester, and Utah are all currently actively enrolling subjects. We have 2 centers that are up and coming, and we really hope that they will be online soon. We'll recruit 51 participants for us at NIH. It will translate to about 18 people that will come to NIH, including international patients. So we do have funding to bring people at NIH and pay for travel, for example. And it's a 2-year study overall. So this is kind of the schematic of the study design, where it is a placebo-controlled trial. So that's where some of the alternative study designs came in handy that I was talking earlier. So we are randomizing 2 out of 3 people to receive ManNAc, 1 out of 3 to receive placebo. Why 2 to 1? Because that tremendously increases our power to detect a significant difference. So we don't need, because we have such a powerful tool in the disease progression model, we do not need as many people in placebo, and we can have more people on the treatment arm. That also allows us to have a smaller cohort overall and have a more limited time frame. So we're looking at 24 months of treatment. Some people will actually go probably to 30 months and 36 months, because it's 2 years once the 51st person is enrolled in the study. And we see everyone every 6 months. We do repeated measurements. So QMA is one of them, very obviously. So we do use the GNE GNEM disease progression model as the main outcome measure. So we're mostly interested in what is the effect of ManNAc on the disease progression model, whereas the previous phases were more safety and muscle staining efficacy. Now we're really looking at the clinical data. We're also looking at questionnaires, reported outcomes, other functional measures, including the 6-minute walk test, the AMAT, so on and so forth. Here are some conditions to be on a trial. I've put the link if you're interested to have all the full condition, but in the summary, people need to be between 18 and 70 years old, have a genetic diagnosis that's confirmed. So that's where all the data that came from studying the variants comes in very handy. And we are still working on able to communicate in English, because all our questionnaires are in English and we cannot provide translation of them. Able to travel. No ManNAc or salicylic acid derivatives in the past 6 months, some safety labs within limits. So we need to not have someone that has too low platelets, for example, or liver function tests that are very high. So we do have some safety measures to make sure that we're not causing any more harm, especially in some domains where we know that ManNAc had previously had an effect, like on liver function test, which was always mild, but we don't want to, on a drug that we've seen in about 20 individuals taken, we are still on the safer side. And one of the important one, which is a tricky one, is the 10 to 65% of predicted strength on QMA. And you may remember when I was talking about the treatment parameter and the disease progression model, where I was talking about the fact that we needed people to be in the most active phase of the disease. That's what that criteria comes from. So it is to be able to address the question of whether or not it has efficacy in a short amount of time, such as 2 years. We need to be in a state where we're able to make a difference in that short amount of time. So obviously, if I had magic wand and I knew that ManNAc was effective and we could give it to everyone, of course, would want to give it even earlier to try to limit disease progression as early as possible. But if we're too early, as in terms of a researcher, we're not able to make a difference. So we do have to have some limitation on that, even though not necessarily what will represent once or if and once drug gets approval, but we do have to address this for the sake of a clinical trial. So I've put some of the clinical contact information. So both me or Dr. Amato, we are the 2 co-PIs on the entire study. Then there's a project manager at NN109, the contact information at Lenian, or all the site coordinators. So I've put the link here. That QR code will bring you directly to our clinicaltrials.gov page, where you'll see all the specific email address to communicate with any of the sites. If there's one that's you're interested in, you want to communicate with, either if it's yourself or a family member or one of your patients, if you're a provider. So we're always happy to hear more. Some sites may say that they're towards more, they already have quite a fair amount of people on their waiting list. Some others do have more availability. So would communicate with them and see what they say. So as a summary, I think that overall, we can say that ManNAc has shown some data on efficacy and muscle silation. We still have ongoing questions to address in terms of clinical efficacy, but we have some encouraging data from the previous phases. And we are very lucky that we had so many people come with the natural history that help us develop innovative new ways of looking at disease progression that help us now address important questions of efficacy and clinical outcomes. So happy to entertain any questions if there's any.
Yeah, thank you so much, Francis, for giving up your Sunday. We appreciate your time very much in summarizing the progress and also the history of GNE studies in collaboration with NIH, the multi-sites, and also with Ledians. And I particularly appreciate how you've highlighted the challenges behind conducting a clinical trial for a rare disease. How that the challenges are very unique to that of common diseases when the cohort is so small, and how to show efficacy in in a drug to treat a disease that's very slowly progressing, that entails some unique challenges in itself. So, and I hope that the audience appreciates that all the work that's been put into by the field, by the NIH, particularly by Marianne's group and those at NIH and all around the world in helping us reach this stage where we can now confidently perform a trial and be able to know if a particular drug had an effect or not. So we have some questions. So Tara's first question is, European group is studying a prodrug for GNEM. Do you anticipate using ManNAc in consortium with this prodrug?
That's a very good question. So we are aware of the GNE group is studying that at this point. So we have an ongoing trial. So we are at this point, we're not necessarily looking to add to. We really need to address, I think, the efficacy of ManNAc by itself at this point. It is going to be a good question if there's multiple treatments and therapies at some point. How will they interact with each other? What is the efficacy of one compared to the other? If we come to the point where we have the luxury of having multiple therapies and having that kind of question, that will be amazing. And I will, luckily, I be like, oh, let's see what we can do. But it is a very good question. But I think we need to gather data on each individual compound before we can try to say, oh, do we need to mix and match?
So Tara has another question. Once ManNAc is approved, how affordable will it be for patients globally?
That's a good question, and it's a hard one to answer because it depends on so many different factors that I'm not necessarily involved with, which involve drug production at this point and maintaining standards and how much analysis needs to be done on the drug and how the packaging ends up like. And so it's, I cannot answer well that question at this point in time. I have no clue on how. It is a small molecule, so it's
I think it's going to be a lot more affordable than a lot of other types of drugs, for example, gene therapy, obviously. And I think the answer is going to differ depending on, the healthcare reimbursement plans and in each country. That's very different from country to country.
Yeah, I come from Canada. I've seen the difference between price tags in Canada and the US, which are tremendous, even though the border is shared all across. And the thing that it does change quite a bit. But yes, it is a small molecule, so I do not expect the prices to be like the tremendous price that we incur for enzyme replacement therapies, for example, and some other genetic disorders, or like gene therapy, which are more single dose or limited number of doses, but much higher price tag. But except for that knowledge from general genetics, I cannot answer specifically for ManNAc.
Okay, we have another question. Question about ManNAc trial. From a genetic point of view, what are the criteria to be eligible for the trial and or future access to the compound, in particular when the patient has a variant of uncertain significance?
Yeah, that's a very good question, and thank you. I see it's Mr. Santorbi answering that question. We've been there's a lot that goes into that. There's the difference between what we're doing for a clinical trial and what comes for the drug once it's approved. So there's a lot of people that we know have variants of unknown significance. That comes from the fact that there's many missense variants, as I was saying. And in most of the times, there's hardly ever that I see two GNE variants and I think it's not GNE myopathy, even if the lab interpretation is variant of unknown significance. And we do have a set of criteria. They're the ACMG guidelines criteria, which has been published in 2015, the Richard and all criteria. That's what we're using for the study. It is more challenge to be able to show to regulators and everyone that we have a cohort that is solid and that we're convinced that everyone has GNE myopathy and we're not intervening on anyone else. Once a drug is approved, then it becomes the responsibility of the individual physicians to decide whether or not they want to administer the drug. And a clinical diagnosis of GNE myopathy, even if the variants are not as convincing as others because we have less data. If the clinical diagnosis is there, we can intervene. It is a very different question in between the clinical realm and the clinical trial realm. So for the clinical trial, we have to convince everyone that we're doing it the more strictly possible. So we have to have highly curated variants. And that's why we're so stringent on it. Once it comes to approval, physicians will be free to do whatever they think. And if they think it's GNE myopathy and they have good evidence of it, even though the variants are not as convincing, there's nothing that will stop them to doing that.
So long story short is basically for the trials, for the sake of the trial and getting convincing data, you're only including participants that have very strong curated variants. Okay.
Just to prevent a couple of and we also have a criteria that we are looking that the lab is approved. So sometimes we do have to repeat the testing. It has been in a lab that is we get a lot of labs from all over the world. So we do have a criteria that it needs to be a CLIA approved lab or a very reputable lab as determined by the investigators. So we're more stringent because some labs may do interpretation and sometimes very surprisingly, like interpretation that go the other way around. So I've seen the very common Valin 727 variant that's most frequent in the Indian population interpreted in some labs as a variant of unknown significance, for example. So depending on the lab don't use all the same criteria, we try to normalize everyone so that some people that do have very clear pathogenic variant, we're not missing on them. At the same time, making sure that we are as solid and we can convince everyone.
So we're close to time, but let me just fit in one more question before we close out. Someone has a question. If we cannot travel, can the ManNAc trial be done with the help of the main hospital in the country, e.g. UK?
Yeah, so really for that study, the answer is going to be no. Like we have multiple standards here. All of them can accept international patients. The main site for international patients has been designated to be NIH just because we do have the funds to help for travel costs. And we do, like when people come from, we've had people from all over the world and we pay for travel. If they need some assistance for travel, we do pay for the person that helps them for the travel and their lodging and some reimbursement for food as well. So we have that kind of budget to try to address that issue, but we do not have any centers internationally, and there's no intent at this point to try to broaden the trial to other countries at this point.
We do have some more questions, but Rachel, do we have time? Is it okay to go over, or should we close out and let Jeffrey give his address?
If you and Francis don't mind going a little long to answer the questions, I'm fine with it.
Do you have any recommendations on how to get improvement on already weak muscles other than taking ManNAc? This is a question from someone.
So that would come more from my experience from the natural history, where we've seen several people that are not on treatment, what we've seen. So it's kind of not using the muscle is deleterious to anyone. So we all know, and there's plenty of data, and not only muscle diseases, but healthy individuals, that the less the muscle is used, the less it is going to perform. So physical therapy, occupational therapy, exercising in a directed manner. So we never want to injure the muscle, and that's what we've seen. And it's not I wouldn't say it's not a strong tendency. It's kind of a word of the mouth that we're observing in the natural history, where people that report more trauma or periods of more intense physical activity, all of a sudden, let's say for work, noted more progression after some of these episodes. So we're kind of careful of suggesting exercising. I always say it is good to have someone to help direct physical therapy, have an exercise program without straining. So we want to avoid muscle injury, but use the muscle enough that it preserves as much strength that it can.
That makes sense. So another question, if ManNAc safety is already proven, can it be sold in the market as a regular sugar drink?
The thing is the safety proven? I would say probably not. So that's the safety is always an ongoing issue. We have safety data on about some people are repeated between phase 1 and phase 2, but basically 20 individuals. So how much, like, I do not have significant concerns at this point about the safety. Over 2 years, given it's going to be a 2-to-1 trial, we'll have 34 people on ManNAc. That is almost double than we've ever seen. Will we see more rare side effects that we've not seen previously? That is the question. So we need to be aware of this, even though we expect, so everyone has some ManNAc in their body, and even with GNE Myopathy, there's some ManNAc that is there. So it's not necessarily expecting very severe adverse events, but we still need to gather more safety data. And we also need to gather efficacy data, because if for some reason we end up in saying, well, we do not see a difference, and we've seen that in other molecules in the past, that the final trial was not necessarily as positive as people were hoping from the data from the previous phases. We didn't want to give something that has some side effects and not having an impactful medication at the end. So we do have the duty, I think, to prove that it really has an effect, that it's really safe. And
Great. And someone else has a question based on a study by Nishino and Noguchi's lab, where they published that sialolactose ameliorates myopathic phenotypes in a GNE model mice. Why the 6-sialolactose has never been done in a clinical trial, given that it's apparently in some baby formula milk?
It's a good question. I know a bit about the sialolactose data, but I think that there is actually a trial ongoing in South Korea of sialolactose. I don't know. I don't remember their exact formulation and the link if it's a 6, but there is some data being collected on sialolactose as well. It not has been the focus of NIH, because historically we've always been more interested in ManNAc because it is uncharged, so we knew that we could get it in the cells more efficiently. So that's why we focused on that molecule at first. But
it could be that the Japanese group are trying that because they were the ones that published on that, but I'm not too sure.
Yeah, I'm pretty sure it's currently the sialolactose is the Korean group, and I think the Japanese trial ongoing is on ace-nuramic acid. If they've not finished since, like, they may have collected all their data by this point, but I know they were. I think that those are the trials that were either ongoing or are now performed.
Okay, one last question. How long will it take for ManNAc to be out on market after the trial is finished?
That's a good question, and it involves many different layers. So it's not only the scientists behind it, and it's not only the patients, it's also regulators. And so it depends on how fast we can put the data together, how convincing the data is, and if there's any type of question. So
it's hard to know because it depends on the FDA's decision, and then it depends on the company leading, how quickly they can mass produce it. There's so many factors, right?
There's so many different factors. And we've seen, like, for example, the FDA approval of the vaccines went very quickly because they had like a lot of data and a lot of stuff. Like, it depends on how, and there's some rare disease drugs that got approved fairly quickly. Some others went a lot slower. So it depends on the kind of data. It depends on who's examining it and what kind of questions are residual after. So at this point, given I don't even have the data, I barely saw a few people on the 6 months visit yet. So we need to wait until we have the discussions at the end of the study, and then we will have probably a better idea of how well it's going at that point.
Great. All right. I think we've gone a lot over time. So Please join me in thanking Francis for educating us this morning, and I would like to pass the stage onto our Executive Director, Jeffrey G, to make some closing remarks.
Thank you both. Francis, very enlightening, and we really appreciate it. Angela, as always, thank you so much for moderating. I just wanted to do a year-end closeout to let everybody know exactly where the organization is. We're now in 50 countries around the world. We have a very large number of patient advocates, all volunteers, who are our ears and eyes on the ground, and it makes it much easier for us to know what's going on in the rest of the world. For example, this call, we expect that I noticed from the people who are participating that there are a great number of people who, for example, will be in Australia or Japan or somewhere else where the time zone is very different. And so we expect them to join us in enjoying the recording, which obviously will be up within a few days. Talking of Leadient, we have a very strong and good relationship with them, and obviously Francis and MAGiNE know that the NIH is an area, a place in which we are much involved, and obviously with Angela and the LEC lab at Yale, there's a lot of activity going on there. We have a tremendous outreach internationally. We have a very large scientific advisory committee with 12 people on it. We have a really good infrastructure. That's the best way of putting it. So when I like to think that NDF is the major organization globally, I can really sincerely say that. We lead the way both in research. We have ongoing studies. We have trials all over the world. We have a lot of trials locally. So internationally and domestically, we are very much involved in everything to do with GNEM. So far this year in the speaker series, this is number 16, which is really a tremendous number. We had hoped to do at least one a month, and now we're accelerating to beyond one a month. And everything is archived on our website. It's really a good opportunity for those that don't know much about us or want to refresh their memories. You can go into the website. I always say a large cup of coffee and a lot of bickies or cookies. It's going to take you a long time to delve and enjoy, but the information is there, and our team has done a really good job. Rachel really has led how we do these things so that it's for the benefit of the world and the community. We also have a lot of international relationships with the international organizations, and that really makes a big difference, so that when we get an inquiry from somebody in a country we don't know, the Ivory Coast, for example, we have a certified patient advocate who's a volunteer there. They feed the information back to us, and then we disseminate it as best we can for the benefit both of the patient and their families. So this really is a year-end pitch. I want everybody to say goodbye to 2022. I think we had enough of that, and to look forward to 2023. I think we have great hopes for the ManNAc trial. We have great hopes for our future, trying to get COVID behind us and everything else that's going on in the world. And I would invite you, if you haven't already done it, to think about a year-end donation to the NDF. It would be remiss of me if I didn't say it. Our website is cureGNEM.org, and Rachel has already put it up. So a simple click will get you to a donation page. Thank you again, Francis and Angela. We really appreciate your involvement, and as always, wishing everybody the very best for the holiday season and for the new year. Thank you.